Hello, my dear kiddos. Hope you all are doing good. So today we are going to see the mock question paper 2024. We are discussing previous year mock for this year, and this is part 8. Let us begin this session. Which of the following is incorrect with respect to aves? Excretory organ is proboscis gland. Presence of four-chambered heart. Presence of feathers and beak. Presence of long bones which are hollow with air cavities. Which is wrong with respect to birds. Your time starts 3, 2, 1 right now. Children, their excretory organ is not proboscis, right? They do have kidneys, right? Yes. If a cDNA is digested by a particular restriction endonuclease, 8 base pairs away from the center of the palindrome sequence, what will happen? DNA fragments with flush ends will be produced. Resultant DNA fragments may not have the overhanging stretches. The DNA fragments will facilitate the action of DNA ligase. The DNA fragments will not have sticky ends. Your time starts 3, 2, 1 right now. Here goes the answer. The DNA fragments will facilitate the action of DNA ligase. Why? Because restriction endonucleases like E. coli cut at specific sites and produce sticky ends, overhanging stretches. These sticky ends are helpful in binding with other DNA fragments using DNA ligase, which joins them during genetic engineering. Okay? Right. Yes. Okay. Basically, the fragments will have sticky ends. But other option says that it will not have sticky ends and not have overhanging stretches. Okay, but here, since it has sticky ends, right, it is helpful in binding with the other DNA fragments using DNA ligase. In 1983, Eli Lilly, an American company, prepared 2 DNA sequences corresponding to A and B chains of the human insulin and introduced them in plasmids of Salmonella typhi, Thermus aquaticus, Medicago, Truncatula, Escherichia coli. Your time starts 3, 2, 1 right now. Yes, in Escherichia coli, right? E. coli was used by Eli Lilly to produce recombinant human insulin. Separate DNA sequence, that is for A and B chains, were inserted into the plasmids and then introduced into E. coli, where they produced and later combined. Which of the following is not a method of diagnosis of a disease? Not a method of diagnosis. PCR, ELISA, radiography, radiotherapy. Your time starts 3, 2, 1 right now. Very simple, right? No confusion. The level of difficulty is super easy. It's radiotherapy. Children, therapy means treatment. It's not a diagnosis, right? Read the following and choose the incorrect statement. Okay, RDNA technology serves the purpose of early diagnosis of disease. RTI could lead to several complications like PID, stillbirth, ectopic pregnancy, cancer, etc. Hepatitis B and HIV can also be transmitted by sharing of infected needles, surgical instruments, infected blood transfusion, etc. Mature insulin has 3 polypeptide chains. Which do you think is the wrong statement? Your time starts 3, 2, 1 right now. Yes. Mature insulin has 3 polypeptide chains, they are saying, That is very wrong because it has only 2 polypeptide, which is A and B, linked by disulfide bonds. Which among the following belongs to class Mammalia? Acidia, Canis, Chelone and Corvus. Your time starts 3, 2, 1 right now. Again, very easy. Yes, the answer is Canis. Canis is nothing but what? Dog, right? Acidia is protochordate, Chelone is a reptile and Corvus is a bird. Select the odd one with respect to secondary metabolites. Concanavalin A, tyrosine, vinblastine, ricin. Your time starts 3, 2, 1 right now. Odd one with respect to secondary, odd one out. Yes, it's tyrosine. So tyrosine is a primary metabolite. Okay, it's an amino acid and it is essential for the growth and development. Other concanavalin A and vinblastine and ricin are secondary metabolites used in defense, signaling or storage and not basic metabolism. During fertilization, a sperm comes in contact with the X layer of the ovum and induces changes in the membrane. That is why the entry of the additional sperms. Membrane is Y. Okay, select the correct option for X and Y. What is X? What is Y? X layer of the ovum and induces changes in the membrane. That Y, the entry. Okay, fine. Okay, so basically they are saying that Y the entry means what does that does to the entry of the additional sperm. Zona pellucida and block, zona pellucida and promote, corona radiata and promote, corona radiata and block. So X and Y respectively is this and this they are saying. What is that layer and what is the functionality? What does that do to the additional sperms? Your time starts 3, 2, 1 right now. Yes, it's definitely zona pellucida. It hardens and it prevents. Once fertilization happens, right? Once one sperm is selected and it fuses in, it hardens and it will not allow other sperms to enter inside. So it is blocking polyspermy, right? Yes. So it binds to zona pellucida and triggering acrosomal reaction and entry. This blocks polyspermy. Yes. A person with which of the following blood group can receive the blood from universal donor only? A, O, A, B, B. Your time starts 3, 2, 1 right now. Okay. It's actually O group, right? O group can receive blood only from O negative, right? Yes. O negative is the universal donor, by the way. Blood group O has no antigen and both anti and anti B bodies, anti A and anti B. So it can only receive blood from O, which makes it the universal donor and not the recipient. Select an autoimmune disorder that affects neuromuscular junction. Very easy. Osteoporosis, myasthenia gravis, Addison's disease, muscular dystrophy, which is affecting the neuromuscular junction. Your time starts 3, 2, 1 right now. Yes, it is myasthenia gravis, right? It's an autoimmune disorder where antibodies block receptor at the neuromuscular junction, causing muscle weakness and fatigue. Right, with that we are completing this session. I hope you all enjoyed the questions. For more such videos, stay tuned and do like, share and subscribe. All the best for your exams. Bye bye. Take care.